All right, guys, today we're going to be looking at our agenda, which is going to be letting you know that today we are working on our own. It's an asynchronous lesson. I wanted to make the point just to show you all a few things in the agenda. So, first of all, there is an optional lecture. So, if you continue to watch the rest of this video, that's where the lecture will be. I'm also going to go over a couple of answers for experiments and ethics. So, I am going to go over a few of those answers right here that will help you in this refinement section. Next class, we will be doing statistical analysis, and we will be hosting everybody during that class period. So, do make sure that you are prepared to participate during the class period. All right, if we click next down here, so that always takes us to the next guy. So this is our lecture, research methods. We are going to be looking at a specific slide. I'm going to point that out to you here in just a second. So I'll be starting this lecture here in just a moment. I would like to point out a few things before I get started on that. I would like for you to make sure you look at your grades tab and you look and see exactly what percentage of the material you've covered so far. So your grade right now is only the percentage is representative of only your completion and correctness. So your accuracy and completion. You'll notice that those are all out of like point values of 1 or 2. So 1 usually meaning it's complete or incomplete, and then 2 meaning it might be more than one page. If you want an easy way to complete assignments, then go to the assignments tab here. So when you pop into the assignments tab, that will show you exactly what you have done. And it'll usually have like a little over here for me, it's a check on if I've graded them or not. And then right here, this tells me if it's synced to Infinite Campus. So you'll notice that I've turned on the syncing for all of the assignments. So today you will see in Infinite Campus your grade appear. So that will kind of open up the floodgates for communication back and forth between not just me and you, but also me, you, and then whoever else is kind of overseeing your grade. All right, next. I'd like to go back to our home. And then once again to our modules. All right, and then flipping down here. So I'd like to talk for just a moment about this target reflection. So I'm going to go ahead and open it up. And so you'll notice that again, we are putting all of the assignments for the day in just this one handout. So it looks like this target reflection. All right, so my suggestion is for you to go ahead and take the quiz and circle your hits and misses first, and then kind of go from there. And then I would probably say, do that first and then come back to this video. So I'd like to type in a couple of answers for these. So 1, 2, and 3. So I'd like to type some of those in so that you guys can just see what the right answers are. And that way you can kind of check for yourself. Do you know if you're on the right track? All right, so I want to remind you independent variables are that's the things that change, and the dependent variable is what we measure. So, of 100 individuals with moderate depression, 50 receive 8 weeks of a new cognitive behavioral therapy, while the other 50 are placed on a waiting list for 8 weeks. At the end of the 8 weeks, all 100 are given psychological tests to assess their level of depression. All right, so again, our independent variable is what changes. So what does one group get and the other group doesn't? And that's the cognitive behavioral therapy. So I'm going to just use the acronym for that. So cognitive behavioral therapy is what changes. So you're either given it or not. And then for the dependent variable, it's what we measure. It's their level of depression. Now, your control group is the group that basically doesn't get the treatment. So the treatment is what the independent variable is. So the group that doesn't get any treatment is essentially the ones on the waiting list. And then the experimental group are those that are given the therapy. All right, let's look at number 2. A biopsychologist is studying the effects of anabolic steroids on the aggressive behavior of female rats. 24 female rats received daily injections of a placebo, a fake drug, while 24 others received daily injections of the steroid. Round-the-clock videotapes of the communal cages of all rats allow all aggressive encounters to be counted and timed. So let's look at independent variables. So again, that's the thing that we are seeing change. So what is the thing that they give the rats? That's anabolic steroids. And then what do we actually measure? Aggressive encounters. So how do we measure that? With counted and timed interaction. The control group is the group that doesn't receive the anabolic steroid. So in this case, when we say anabolic steroid, the group that doesn't get it is a word that we learned when we took our notes to prepare for this class, and that's the placebo group. So those are the people, sorry, not the people, but the rats receiving placebo. And the rats who receive the steroid are those that are in the experimental group. All right, I hope that that helps you. If you still have some questions and you'd like to get a little reminder over what these kind of terms mean, then I'd like for you to stay on here and just continue to watch. If you're feeling really comfortable and confident moving forward, then it's time for you to go ahead and pause this video and go ahead and move. For the rest of us, so let's go back to this lecture. So I'd like to talk for just a little while, just to give you a little review of some of the terms that you learned about in the experimental and ethics lesson. All right, so you remember this is the PowerPoint that we used last class. So it's all the same, just so that way there'd be one file for you to look for. So I need you to go ahead and scroll down to slide number 37 if you're trying to follow along with me. And that is where we start experiments and ethics. So the first thing is I want to go back to the modeling system for how we are going to remember this material. So you should already have sort of like a schema laid out in your brain, a centrality model of all the things you associate with a survey. These are all the things we looked at and learned about last class. So population, sample, random sample, representative sample, a correlational coefficient, and causation is not correlation. Those are things that you should already know now. And then you should also know that in an observation, we sometimes have participant or observer bias. And then we also know that case studies have strengths and weaknesses, the strengths being that we can get in-depth information, the weakness is that we can't generalize it to the public. So today we're going to be trying to make sure we talk about all of these, so our experimental information here. All right, so what we do know is that we need to remember what experiments are and what they aren't. So they aren't correlational studies. That means we can, in fact, draw conclusions about causation there. We know that they are not surveys, right? We know that they are not some other terms that you learned about last class, longitudinal studies, cross-sectional studies, or cohort sequential studies. So what they are is they are containing an independent and dependent variable. The independent thing is what we manipulate, and then the dependent being what we measure. We divide and control those our population into control and experimental groups. And then we have to use random assignment in an experiment. That's where we randomly place people either in the control or the experimental group. So we're going to go ahead and talk about some of those terms. So again, just to remind you, so we talk once again, our independent is the thing you change. We usually limit it to only one in an experiment. Sometimes we can have more than one independent variable if it's a complex one. And then our dependent is, again, what we measure. And then our controlled is everything we want to remain constant. So the controlled variable is, in this particular example, is the plant itself. They're not using all kinds of different plants. It's the same exact type of plant each time. And then they are just changing what they water it with, right? So we need to make sure we know exactly which variables are independent and dependent or just controlled. Sometimes it's hard for me to, it used to be hard for me to remember independent versus dependent variables. So I always try to come up with like a wild explanation for myself because that is what your memory kind of grabs onto most easily. And I always think in this case of this song, Independent Women, just a little refresher for those of you who might not have heard it before. It's a banger. Lucy with my girl Drew, Cameron D and Destiny, Charlie's Angels, come on. All right, obviously, it's just going to be incredible. So the way I kind of remember or visualize independent and dependent variables, and then I host this in a place in my brain which is in charge of remembering faces, which is a really strong part of everyone's brain. I think about Beyonce and I think about her husband Jay Z, and so I start thinking about what are some things that change, and then what can we measure as as how a change is affected. So I think of her like kind of in the single ladies days, and then I think of how she's changed in the lemonade times, and sort of how those two different women and those two different people that she's been over her lifetime have maybe impacted her husband. This is Jay-Z. So I just think of I'm going to measure Jay-Z's reaction and what he acts like, and my independent variable being what changes. It's going to be her kind of attitude and her gestures towards him. It's just a kind of funny way to remember the word independent, right? The one that changes. So I think of her as like this independent woman, and then dependent, the one that's measured for effect. If that's helpful to you, hooray. If it's not, then you just pretend like I wasn't even talking. All right. Another thing that we do, so once we've established our independent and dependent variables, we need to determine what kind of population we want to gather information from. In psychological studies, oftentimes the population is a rat. And that's because rats' brains and humans' brains are very similar. Especially in drug trials, we always want to use rats first. And that's just to, we'll talk about ethics here in just a second. So we have our population. Now, we remember from last class in our survey discussion that we always want to have a representative sample of the population. So, what do we do once we have a representative sample of the population? Well, when we experiment, obviously we randomly select them into this group. So, this random selection happens. We have the population. We randomly select people into this sample. And when we say random selection, remember it's a representative sample of the population. You'll notice that it has the equal number of blue dots and yellow dots. And then what makes an experiment special is the random assignment bit. So then we take that sample and we randomly assign those into the control and the treatment groups. So we essentially can use any number of tools to randomly assign groups. So that's what random assignment is. Oftentimes students get random selection and random assignment separated in their brain somehow, or they kind of actually make them go together. And they are two very different things. So, random selection is taking from the population and getting a representative sample. Random assignment is the control and the treatment groups. And we sometimes call this experimental group. Why do we use random assignment? Well, the reason why we use random assignment is we want to reduce the number of confounding variables. So, if we have a group of humans that have all sorts of different walks of life, but we accidentally, without knowing it, assign the experimental group, in this scenario, like all women into the treatment group and all men into the control group, well, then we have a whole big problem because now we don't know if the reason why the experimental group shows a difference is because of their because of their gender, or if it's because of the actual treatment we gave them. So that's problematic, right? Now, we also want to be able to allow for causation to be determined. So that's another reason why we use random assignment. We want to make sure we take out the confounding variables, and then that allows us to assign the causation from just the independent variable alone. All right, now there's another part of the experiment you have to decide on, and that is, are you going to use a blind? So, what is a blind? So, let's say that you're the doctor, right? So, this is an experiment, and as the doctor, you are giving your control group and your placebo group pills. These groups don't know which group they're in, so they don't know if they are getting actual medicine or if they're getting a placebo. And we want to do that in the case where we are not really sure if the pills are actually um, relieving any sort of symptoms, right? So we want to use a placebo in this. So it's good to do that so then the patient doesn't know what exactly to expect. We can then determine, is the actual pill having any sort of effect? Is there any placebo in the pill? Those sort of things. Now, there's another option, which is a double blind. Now, sometimes what we have is if we're the person who's giving out the treatment, can sometimes unintentionally sway the expectation of the patient. Um, so if they do that, then perhaps the group who is in the, you know, test group knows they're getting the placebo, or the control group knows they're getting the actual pill. So if we don't tell the experimenter exactly who's in what group, that's called a double blind, and that's really helpful for a lot of reasons. So why do we use them? So one of them is to measure the placebo effect, but also we want to use a double blind to reduce experimenter bias, so that we don't have any sort of issues that come along with that. Now, we have some other issues that you were introduced to. One of them was the Hawthorne effect, which is that if you know you're being observed, then you start to act differently. So, that's something that if you're in an experiment and it's in a lab scenario, we can't always get around that. Sometimes we just have to account for it. We just have to say, hey, one of the problems with this conclusion is they knew that this was going on. And so that might have changed their reporting. The only way that we can really get away from that Hawthorne effect is usually using some sort of naturalistic observation. And that's in the category of correlational studies or descriptive studies. That's not in the experimental study. All right, if you need a review, there's a great review on research and experimentation on the Crash Course site on YouTube. I'm not going to play it here, but great review if you still need some help. Now, ethics are another subject that we tackled. And our best example is this a way to remember it for our ethical guidelines in psychology is this acronym DDRIP. So DRIP. And I love that people are saying respect the drip. Now that might be helpful for you to remember this. So I'm going to play this for just a moment so you guys can kind of see what these mean.
Hello students, and welcome to the next lesson in our AI Psychology course. Today we're going to be looking at ethical guidelines. What are ethical guidelines? After the Stanford prison experiment, which was conducted by Zimbardo, which looked at how social roles are going to influence behavior, the British Psychological Society came up with a set of guidelines for future psychological research. This was done to ensure that all psychological research is in fact ethical and does not affect the participants in any way. So what were they? They were classified as DDRIPP guidelines, or otherwise DDRIP. This is an acronym which stands for all of the names of these guidelines. The first of which is deception. Participants must be told more or less what they're going to be doing so that it doesn't completely deceive them when doing the experiment. This could then make them feel bad after the experiment has taken place if they haven't actually being told completely what they are going to be doing in this experiment, and therefore have been deceived. So it's not like you have to tell them every single detail of the experiment, because if it's an experiment which is going to test their reaction to something, you don't want to pose a reaction post the pre-experiment which is going to make them want to react in a certain way. But you have to tell them more or less what is going on. So for example, in Milgram's experiment, people weren't actually told that the participant is actually a Confederate and fake, and screams and the kind of way a participant is being hurt when subject to that is in fact fake, because that would completely throw it off. But in Milgram's experiment, they were completely deceived, and that could actually harm them. Milgram's experiment is a big experiment which we're going to be looking at later in the course when we look at obedience. So, debrief. This is a guideline where participants must be told that they have the right to remove themselves or any data from the study, and they're debriefed from their kind of rights in this actual experiment. They are also debriefed and told that they have the opportunity to talk to the experimenters to know the true aims of the study. So this kind of debrief after the experiment allows them to know any information which they didn't know prior to it, and therefore makes them feel a lot better about what they've just gone through. Right to withdraw. In this instance, the participants are told that they can remove themselves from the study at any time they want. They are told how to withdraw and how not to be harmed when withdrawing, so as a result, if they're actually doing the experiment, they're not going to be harmed. For example, in Zimbardo's prison experiment, they weren't really given the right to withdraw at all, and as a result, the people in the experiment suffered psychologically in terrible instances. For example, the prisoners who actually had to be removed from the experiment physically because they were being psychologically traumatized. If they were given the right to withdraw, when they did feel slightly traumatized, they could have just left and prevented a lot of psychological harm. Which brings us on to informed consent. Participants may only participate in an actual experiment if they've consented to take part in the study and they know all the information involved. So this pretty much means you cannot participate in a study if you haven't given full consent, knowing what's going to actually be happening. If you don't, then therefore you are breaching the guidelines. Protection from psychological harm. In this instance, participants must be managed so that they can exit the study unharmed and they're not traumatized. They are made sure that they are leaving unaffected. So this can be perhaps a debrief from psychologists, which can then let them know how to actually interpret everything they've been going through, and then as a result, they're not going to be harmed psychologically when they leave. Privacy. This is a final guideline, and it states that participants have the right to know who is going to read their results in a report, and they also have the right to remain anonymous and make sure that their life remains private in the experiment and their name isn't publicized. Researchers also can't invade their personal life when conducting the experiment because they have the right to remain private. Here's some questions on the ethical guidelines or the DRIP guidelines. Have a go at answering these on another sheet of paper by pausing the video and then hit play for whenever you're ready to see the answers.
All right, so that's our kind of nice little ethical guideline answer. Not answer, but review. And it's really important that you understand just what they are, right? So just the answers, just the acronym, DRIP. So that's really all that you need to know about these guidelines at this point. Over the course, we will be doing quite a bit of practice with ethical guidelines, and I'll be giving you lots of scenarios where you are going to have to determine which of the ethical guidelines are being or aren't being met. All right, so that's it. I just want to make sure that you move on here. So make sure that if you haven't done so already, you do take the quiz. So this is on Google Forms today. And then do make sure that you go ahead and complete the target reflection once you've finished that. So it shouldn't be too much longer. All right, thanks. Have a great day, guys.